Hello, in this short video we will be going over what protein synthesis is and how it is done within a cell. So let's get started. So what is protein synthesis? Protein synthesis, also known as protein biosynthesis, is a process where organisms create new proteins, and this takes place in the nucleus and the cytoplasm of a cell. Proteins are very important as they make up most of an organism's body. They are made of amino acids and vary in functions from maintaining your body, being sources of energy, creating enzymes and antibodies. Without proteins, organisms cannot survive, and thus is why protein synthesis is so important to living organisms. Before we begin explaining protein synthesis, let's go over what RNA is and why it's so important. rRNA stands for ribonucleic acid and is a copy of a segment of a DNA. Its main purpose is to be used in protein synthesis to create specific proteins. There are three types of RNA, and they are messenger RNA, ribosomal RNA, and transfer RNA. mRNA carries genetic information out of the nucleus and into the cytoplasm. RNA can be thought as a cell machinery that builds proteins. Lastly, tRNA brings amino acids to the ribosomes and are then assembled. They have an amino acid attached to its end, and on the other end is a series of specific nitrogenous bases that correspond to the mRNA called the anticodon. There are 20 different amino acids, and they all have one specific tRNA that corresponds to it. Now let's get on to the steps of protein synthesis. There are two steps to protein synthesis: transcription and translation. Transcription is the step where a particular segment of DNA is encoded into mRNA, which it then travels out of the nucleus and into the cytoplasm. Translation, on the other hand, is the step where the mRNA is read by a ribosome and then tRNAs come along and produce amino acids accordingly. Let's take a look at transcription in more detail. Transcription is also known as RNA synthesis, and it takes place in the nucleus, as that is where the DNA is located at. The purpose of this process is to create mRNA, which carries partial genetic information, which is done by RNA polymerase. There are three steps in transcription, and they are initiation, elongation, and termination. In the initiation stage, DNA is unwound by an enzyme called helicase at the promoter region, and one of the strands will become the template strand, also known as the antisense strand. During the elongation stage, RNA polymerase moves along the template strand in the direction from 3' to 5' and synthesizes an mRNA. This would be complementary to the template strand. 3' and 5' are the ends of a DNA sequence, and this allows scientists to get a sense of direction. Finally, in the termination stage, mRNA and RNA polymerase break away when they reach what is called a termination signal. Now let's take a look at translation. Translation is a step that is involved in actually synthesizing proteins, which takes place in the cytoplasm where ribosomes are located. This step begins with the tRNA attaching to the mRNA. tRNA will move along mRNA, and once it reaches the start codon, it begins synthesizing proteins. This is done by reading the codons, which come in 3 letters at a time, and a corresponding protein would be made by the tRNA. This is repeated until tRNA reads the stop codon, which indicates the process to stop. The amino acids are bonded by the ribosomes as it is synthesized by the tRNA. The bonded amino acids will then be called a polypeptide chain. Now let's take a look at polypeptide folding. Polypeptide folding is a process in which a polypeptide folds into a specific, stable, functional, three-dimensional structure. This could occur as early as translation stage, which takes place in the cytoplasm. There are 4 types of structures when it comes to polypeptides, and they are primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure describes the structure of an amino acid when it is covalently bonded in a single file. Secondary structures are caused by helices made from polypeptides bonding to other polypeptides, and pleated sheets, which are polypeptide strands aligning parallel to each other, and these form due to the hydrogen on the backbone of the amino acid forming hydrogen bonds with the oxygens of other amino acids. Tertiary structures are caused by the bonding of R groups of the amino acids. The R group of the amino acids are sometimes hydrophobic, meaning they're scared of water, causing them to clump together, bending the polypeptide chain. They're also hydrophilic, water-liking R groups too, which could form hydrogen bonds with other hydrophilic R groups within the amino acid chain, resulting in again bent polypeptide chains. Quaternary structures are formed when different chains of amino acids form bonds with each other, creating one big structure. Thank you for watching.